previous times?
No, that's okay. Good morning, everyone, and welcome to the Wake Research Institute webinar series. We're now solely a webinar due to the refurbishments in Charles Walker and the COVID restrictions. This morning, I welcome Dilrukshi, who is a postdoctoral research fellow working with Vinay Pagay in this building, and today she'll be talking about can dry farm grapevines adapt fast to future dry climates.
Thank you, Rebecca, for the nice introduction, and thank you very much, everyone, for coming along to the webinar. Today, I would like to talk about the work that we did to understand how dry farm grapevines adapt to long-term drought stress. So this is not just my work. There are a lot of people who has made a lot of contributions to this project. The main work was done by Tarita Furlan as a part of her honours project. So before going into the details of this research, I would like to give you a brief introduction. So as you all know, drought is one of the major factors that detrimentally affects all crops, including grapevine. So in Australia, grapevine is mainly grown in southeastern part of the country, which is experiencing more unexpected drought and heat wave events. In this graph, on the left, it shows the anomalies in rainfall in southeastern Australia over the last 120 years. This red columns represents the years that had rainfall deficiencies. And the graph on the right shows you the temperature anomalies over the period, and it also shows you the blue lines are the sea surface temperature and orange lines are the air temperature. And here you can see that over the last 2 decades, we have experienced more drought and heat wave events than ever before. And because of the most recent extreme weather conditions in 2019, Australian grapevine production was reduced by 20%. And these images shows you the projections of winter rainfall and temperature in 2019 based on the climatic simulation models. What this means is these images indicates that Australia is likely to have low winter rainfall and is likely to increase temperature by 2 to 2.4 degrees by 2019. So these kind of extreme weather patterns can detrimentally affect Australian grapevine production. Therefore, we have to have some strategies to minimize damage to the wine industry. So one of the strategies to identifying drought-tolerant cultivars and clones that could perform better in dry climates. So over the past decade, we have learned a lot more about how plants respond to drought stress. And now it is very well known that roots sense subtle changes of the soil moisture. And when soil dries out, roots produces some stress signaling molecules such as abscisic acids. And these stress signaling molecules translocated from roots to the upper parts of the plant to initiate all these drought responses. And particularly stomatal closure is the main drought response to minimize the transpirational water loss. But the downside of this stomatal closure is that it reduces the carbon dioxide intake and therefore it decreases the carbon assimilation. Because of this lowering photosynthetic capacity, plants produces a lot of reactive oxygen species when they have higher light intensities. So plants have efficient mechanisms to prevent this kind of the photo-oxidative damage from the reactive oxygen species, and they can activate these antioxidant systems. And in addition to that, they also can adjust their osmotic potentials by increasing compounds like proline. And so in addition to these short-term responses, plants also can adjust their plant architecture when they experience long-term drought conditions. For example, they can reduce the leaf growth. They can adjust the leaf architecture to minimize the transpirational water loss, and also they can maximize the root water uptake by increasing root to shoot ratio. But in terms of grapevine, we know a lot more about short-term drought stress responses, because many of those studies have been conducted by exposing grapevines to a single drought event. But perennial crops like grapevines, which grows for many decades, undergo many recurrent drought episodes throughout their lifetime. So, but we still don't understand most of the physiological and molecular mechanisms that allows them to adapt to long-term drought conditions. So one of the fascinating things that turns out in other perennial crops is that when they expose to long-term drought stress, they have indicated that they can remember those stress events by using their stress memory. So this stress memory helps them to adapt faster to the next stress event. So in some of the studies, they have also shown that this stress memory can transmit to their offspring. But still, we don't have any idea about the existence of this kind of transgenerational priming in grapevine. And therefore, we were really interested to know whether dry farm grapevines, which grown under multi-decadal droughts, can adapt faster to drought events. To answer these questions, we characterize some dry farmed Cabernet Sauvignon clones grown in Coonawarra region, and these dry farm clones have been grown without any irrigation over 65 years. And this vineyard was established as a mass selection. So these clones have unknown origin and unknown clonal identity. So what I'm referring as clone is that population of vines derived vegetatively from a single mother plant. So even though initially they are genetically and phenotypically identical to each other, due to accumulation of spontaneous mutations over time, they can generate some phenotypical characteristics. So some of these phenotypic variations might be really important in grapevine improvement programs. So the overarching aim of this project is to identify the superior drought-tolerant dry farm clones. And main questions we wanted to ask was whether these dry farm grapevines are more adapted to drier climates, or if so, whether they can transmit their drought adaptation traits through their vegetatively propagated progeny to make them more drought tolerant. So the main hypothesis we established at the beginning of the research was if dry farm clones are adapted to drought, they should be able to maintain favorable plant water status, induce stomatal closure to minimize the transpirational water loss, and they should be able to improve photosynthetic performances and also leaf level intrinsic qualities efficiency. So intrinsic qualities efficiency is defined as the rate of net carbon dioxide assimilation rate to stomatal conductance, and this is one of the main important trait for breeding. So the second hypothesis was if those drought resilient traits are inheritable, the progeny also should exhibit some improved drought adaptation. So, so the initial field selection of these clones were done in 2018 in Coonawarra. So Coonawarra has a Mediterranean climatic conditions, and the mean general temperature is around 19 degrees, and it has winter rainfall around 560 millimeters. And terra rossa soil is the predominant soil type, and it is a very porous, well-drained red clay loam soil. And underneath this terra rossa soil, there's a limestone sublayer, and this soil depth to the limestone sublayer is quite variable in this region. And one of the parameters that predominantly affecting vine performances and vigor is this soil depth. So therefore, we initially selected 16 superior vines by characterizing thousands of clones in the vineyard. 16 vines were selected based on their canopy vigor and yield, and they were all negative for UTIPA and leaf roll viruses. And then to see whether this soil depth has an influence on the vine performance and drought adaptation, we categorize these clones into two different categories. And shallow-rooted clones had a very restricted root growth up to 28 centimeters, and deep-rooted clones had average rooting depth of 78 centimeters. And we analyzed soil and plant water status and leaf gas exchange parameters. So in the second experiment, we vegetatively propagated the cuttings from those clones in the Yalumba nursery. So 5 vines were propagated per clone. Out of these 16 vines, we were able to successfully propagate 7 shallow-rooted and 2 deep-rooted clones. And they were all grown in under glasshouse conditions. And then we assessed them under 2 different cyclical drought conditions. So here in this in graph, you can see the soil moisture level. We maintain it at 4% soil moisture at both drought stress events. And we measured so many parameters at 4 different time points before exposing them to the drought stress at the first drought event, after rewatering, and at the second drought event. We measured all of these parameters, soil moisture, parameters related to the hydraulic regulatory mechanisms and accumulation of the xylem, ABA, gas exchange parameters, and antioxidant capacity and GABA concentration. We used soil moisture sensor to measure the soil moisture, and xylem AB concentration was quantified by using LCMS. We used LICO to measure the gas exchange parameters, and antioxidant capacity and aquaporin expression in the leaves were measured, quantified by using quantitative real-time PCR, and GABA concentration was measured by Acuity UPLC system, and we used the pressure chamber to measure the midday stem water potential. And let's move on to the results. In field selection, we first looked at the pre-dawn water potentials. And pre-dawn water potential indicates the availability of the soil moisture. And then we looked at stem water potential. We measured it during midday, and it indicates the plant water status. And these pre-dawn water potentials were significantly lower in shallow-rooted clones. That indicates that the shallow soils had much lower soil moisture compared to the deep soil. But interestingly, shallow-rooted clones were able to maintain similar plant water status as the deep-rooted clones. And then when we looked at the stomatal conductance, shallow-rooted clones had significantly less stomatal conductance, and this indicates that those shallow-rooted clones might have very tight stomatal regulatory mechanisms in response to soil drying. And but they were able to maintain the similar photosynthetic rate relative to deep-rooted clones. And finally, the intrinsic water use efficiency was significantly higher in shallow-rooted clones. So, so these changes in the intrinsic water use efficiency is basically maybe due to the significant reduction of the stomatal conductance. So that was very encouraging results, and this was really interesting. And we wanted to look at those mechanisms in further details. And then, so the next question we want to ask was whether these traits are inheritable to the subsequent progenies. So then we did the glasshouse experiment. Here I'm showing the results of average values of 3 best shallow-rooted clones and 2 drought-rooted clones. So in those soil moisture content, we can see quite significant differences between clones. Deep-rooted clones depleted soil moisture slowly compared to the shallow-rooted ones at the first drought cycle, but at the second drought cycle, we couldn't see much differences in soil moisture variations. And again, the plant water status of the shallow-rooted and the deep-rooted clones were quite similar at these D1 and D2 drought cycles. And photosynthetic rate was significantly reduced in both clones at D1, but at the D2, shallow-rooted clones had significantly higher photosynthetic rate. And intrinsic water use efficiency was significantly higher in shallow-rooted clones at both D1 and D2 drought cycles. So this might, so the increase in photosynthetic rate might be one of the reasons to increase in this intrinsic water use efficiency. Here you can see an improvement of the photosynthetic performances and intrinsic water use efficiency in the shallow-rooted clone at the second drought event. So when we look at the stomatal conductance, it was significantly lower in both clones at D1. But when they exposed to the second drought event, shallow-rooted clones showed significantly higher stomatal conductance compared to the deep-rooted clones. So an ABA concentration in the xylem sap was significantly increased in both clones at two different drought cycles. And so previous studies have shown that the ABA accumulation in the leaves induces the stomatal closure, and this clearly shows the correlation between the stomatal conductance and the ABA. And then we measured the mesophil conductance. Mesophil conductance is the carbon dioxide diffusion from substomatal cavity to the carboxylation site. So this is an important parameter that indicates non-stomatal limitations for photosynthesis. And this is also really interesting parameter that influence the intrinsic water use efficiency. So here you can see the stomatal conductance, mesophil conductance was quite similar between clones at D1, but at D2, it was significantly higher in shallow-rooted clones. And electron transport rate between PS photosystem 1 and photosystem 2 is another important parameter which determines the photosynthetic efficiency. So ETR was significantly higher in shallow-rooted clones compared to the deep-rooted clones at both drought stress events. So this indicates that the photosynthetic efficiency was much better in shallow-rooted clones, and we can see an improvement of the photosynthetic efficiency in the shallow-rooted clones at the second drought event. So this also explains the higher photosynthetic rates and water use efficiency that we saw in shallow-rooted clones under second drought cycle. So finally, we measured the aquaporin expression levels. Particularly, I was interested in TIP11 and TIP21. So these are, so aquaporins are membrane proteins that regulate water transport across cell membrane. And so this tonoplast intrinsic protein, the TIP1 gene expression was, so previous studies have shown that TIP21 expression is highly correlated with the hydraulic conductivity. And TIP11 is also really important to increasing the water transport in cell membranes. So, so TIP11 expression was significantly increased. So in shallow-rooted clones compared to the deep-rooted ones. And when they exposed to the second drought event, again we saw significant improvement of the TIP11 expression, but in deep-rooted clones it was significantly down-regulated. So TIP21 expression was significantly higher in shallow-rooted clones at D2, And this might indicate that the water transport capacity of the shallow-rooted clones are much higher than the deep-rooted clones. And overall, we have seen that shallow-rooted clones are much better than the deep-rooted clones in terms of their physiological and molecular drought responses. And also because the shallow-rooted clones have improved performances at the second drought event. So this indicates the transgenerational stress priming. To confirm whether these shallow-rooted clones actually have this transgenerational stress priming, we measured the concentration of a GABA in leaves. So GABA is considered as a marker for transgenerational stress priming. GABA is a gamma aminobutyric acid, which is a non-protein amino acid. So previous studies have shown that the GABA is accumulating at the onset of the stress in plants, and they have shown that this GABA accumulation helps priming the plants to the next stress event. So in here you can see, so the GABA was increased 12-fold in shallow-rooted clones at the first drought event, but in deep-rooted clones there was like 2-fold increase in the GABA concentration, but we couldn't see any GABA accumulation at the second drought event. So based on the previous literature, we think that this might indicate a priming of those plants by GABA. So finally, I would like to conclude by saying that we have seen dry farmed shallow-rooted clones are more drought adapted than the deep-rooted clones, and we have seen from our results that shallow-rooted clones can transmit the drought tolerance traits to their vegetatively propagated progeny, but we would like to further characterize those clones, the superior clones, and one of the objectives for us is to establish clonal identity of the superior shallow-rooted clones, and also we would really like to understand the comprehensive set of molecular mechanisms that helps this transgenerational drought priming. So with that, I would like to conclude the presentation. I would like to acknowledge my supervisor, Dr. Vinay Page and Tarita Phil, and our honor students who did majority of this work, and also Sunita Ramesh, and Edwards Everard, and Anita at CSIRO, and Kat Kidman, and Yalumba Nursery for their support during this project. Thank you very much.
Thank you, Dilrukshi. If anybody has any questions, can you please type them into the Q&A box for us, please? Yeah. Oh. Joanna, if you want to ask a question, can you type it into the Q&A box, which is at the bottom of your screen, please? Do you have a question, Vinay?
So, Dilrukshi, what about epigenetics as one of the possible mechanisms that may confer this transgenerational
Yes.
drought tolerance?
Yeah. So one of the molecular mechanisms that is the previous studies have shown that epigenetic modifications such as histone modifications, methylation, pattern changes, that all contributes to the transgenerational stress priming. And we haven't actually looked at those genetic modifications yet in those clones, but we would like to explore that in more detail in future.
And in the future, will you be looking at any more recurrent in terms of Chief and CR or
Yes.
further aquaport work?
Yes, we would actually like to characterize some aquaporins in different tissues in those vines. So particularly root aquaporins might be really interesting because we have seen a lot of differences between those clones in response to soil depth, and then the aquaporin might be a really important regulator that determines the hydraulic conductance from roots.
So all that aquaport data was just from mates.
Yes.
Okay. Okay, and we have another question. Are the shallow-rooted plants actually still more shallow-rooted in pots, or do they have the same root depth systems as the deep-rooted?
Now, in the glasshouse experiment, we grew all plants in the same volume pots. So they didn't have differences in soil depth in the glasshouse experiment, but what we wanted to look at was whether they have climbed already in the field, and whether we can increase, we can see that improved drought tolerance when we replant those materials in different conditions. So therefore, we didn't change the pot size. We wanted to give them the same soil volume, and then we measured the drought stress responses.
And another question from Zhu Gong. Did you measure the water uptake from the field experiment?
We didn't actually measure the water uptake from the field experiment. It was, so no, we didn't measure that.
Okay. Okay, and a question from Ian Searle. Stress memory appears to involve histone modifications, both removal of repressive histone marks and acetylation to facilitate picture transcription. Post-transcriptional regulations, for example, RNA modifications, also seem to play a role. Have you measured either histone modifications or RNA modifications?
No, we haven't looked at any those mechanisms yet, but those are really important comments, and we would like to explore that in more detail in the future.
Okay, if we don't have any more questions, I'd like to, hang on, one more question. Is there any root growth from the limestone layer? Did you see any biopores in the limestone?
We haven't actually, we didn't actually look at the variation of the root growth because it was really hard to characterize root growth and the distribution of the roots in the vineyard. So therefore, we didn't actually look at the root growth or any characteristics related to the root structure.
Okay, and a follow-up question from Penny Tricker. So if the roots are the same, how is soil moisture depletion difference happening? Are plants bigger?
No, there was no any differences in the plant size, but what we are hypothesizing is that in the glasshouse experiments, we grow them in the same soil volume, but they have shown different soil moisture depletion, but their stem water potential was quite similar. So therefore, we hypothesize that these shallow-rooted clones might have higher water uptake capacity compared to the deep-rooted ones. So, so we haven't actually looked at aquaporin expression in the root yet, but that is something we would like to investigate in the future.
Question or comment from Rob Murray from Soil Science. That was a very interesting talk, but I have to say I don't know much about that area of plant science. In a study of vineyards in the Barossa, excuse me, Mildura and Griffith areas, we found that in almost every case, vine roots were severely limited by poor subsoil conditions and were very reliant on irrigation. The conclusions were that a lot of soil improvement is needed, so your work is much needed. Thank you, Rob.
Thank you very much, Rob.
Okay, if we don't have any more questions, I'd like to thank you for joining us this morning, and thank you very much, Dilrukshi, for presenting her work. So thank you, everyone.
Thank you.